Hi everyone, we're group 13 and our presentation will be about the rational design of selective and bioactive inhibitors of the Mycobacterium tuberculosis proteasome. And this research was conducted by Dr. Jason Sello. So we'll have an introduction where we will introduce the researcher, Dr. Jason Sello, and his experiment. We'll also go over Lewis structures and resonance and discuss how these structures are very important to the experiment itself. We'll also have hybridization, pi and sigma bonds, where we'll discuss how these bonds affect the molecules and therefore the experiment itself. And then we'll end with the summary and conclusion where our chemical structures are discussed and also explain how this experiment is relevant for the rest of society and also chemistry. So on to the introduction. So Dr. Jason Sello is a professor of pharmaceutical chemistry at UC San Francisco and also a researcher at Brown University. He received a bachelor's in science in biology from Morehouse College in 1997 and also a PhD in biophysics in 2002 from Harvard University. Dr. Sello's doctoral research was in diversity-oriented organic synthesis, and it was carried out in Professor Stuart L. Schreiber's lab in the Department of Chemistry and Chemical Biology. Dr. Sello has also performed postdoctoral research in enzymology with Professor Christopher T. Walsh and streptomyces genetics with Professor Mark J. Butner at Harvard Medical School and the Johns Innes Center in England. Currently, Dr. Sello is using experimental methods from chemistry, molecular biology, and biochemistry to develop new antibacterial agents and technology platforms for bioenergy. So some introduction to the experiment. Mycobacterium tuberculosis, or MTB for short, is a deadly bacterial pathogen, and it's important to research it because as it increases antibiotic resistance, it becomes more deadly and difficult to treat. So the MTB attacks the lungs, and the immune system response to MTB is to attack it with nitric oxide. However, the MTB has an enzyme, which is enzyme 20S, which protects the MTB from this nitric oxide. But without this enzyme and without the protection, the bacteria will have no protection against the nitric oxide and it won't function properly. Therefore, we want to try to inhibit the expression of the 20S enzyme. But the Mycobacterium tuberculosis uses proteasome systems for targeted protein removal. And the 20S core particle of the proteasome in M. tuberculosis is a promising drug target, which uses the process of proteolysis. In Jason Sello's experiment, he designed and synthesized selective inhibitors of MTB20S based on its unique substrate preference and the structures of substrate mimicking covalent inhibitors of eukaryotic proteasomes called syringulins. And it's important that these inhibitors are selective in targeting the MTB20S instead of the human counterpart H20S. Because these syringulins don't inhibit the growth of human cells, they may be promising to the tuberculosis drug development. And as you can see in the MTB proteasome, it's a large protein complex, and we can try to bind inhibitors to the complex, but it's important that they are selective in binding to the MTB20S and not the human H20S. The figure shown on the slide is the illustration of two different structures of syringulin. The one on the right is SIL-B, and the one on the left is SIL-A. And these molecules are important because they are the key to the experiment as Sello used to test how they can stop the development of the Mycobacterium tuberculosis. The two synthesized inhibitors, SIL-A and SIL-B, weakly inhibited the H20S, and they preferred inhibiting the MTB20S about 74 times more. So in this figure, SIL-A and SIL-B are both drawn in their skeletal structures, and this is used to simplify the Lewis dot structure without having to convey all the lone pairs and all the atoms. As for very complex molecules like these, the figure may become very busy and difficult to understand. So although not every atom in this Lewis dot structure is labeled, every intersection is a carbon atom bonded to as many hydrogens as it needs to have 4 bonds. So in this case, this intersection, it's one carbon atom bonded to two hydrogens, while in this case, it is one carbon atom bonded to just a singular hydrogen. And the bonds indicated by these wedges and these dotted lines are used to emphasize the geometry of the molecule. The darker bonds, the wedges, come out at us at the screen and they're not planar, while the dotted lines stick out behind the screen, and they are also non-planar. And these are important because they help indicate the molecular geometry of the molecule. So in both molecules, Sil A and Sil B, the formal charges are reduced to 0, and each atom has a full octet excluding hydrogen. And the Lewis structures for Sil A and Sil B are different because Sil A has an extra double bond, while Sil A has a single bond instead of that double bond. And these two are fundamentally two different molecules and not resonance structures because Sil B has an additional 2 hydrogen atoms in absence of the double bond. So here, these carbons are attached to 2 hydrogens, while the carbons here in Sil A are attached to only 1 hydrogen. And so there are 2 additional hydrogen atoms on Sil B. The molecular formula of Sil A is C24H39N5O6, while Sil B is C24H41N5O6. And we can see how the additional 2 hydrogens are accounted for in the molecular formula. And the presence of this double bond makes the bond length between the carbon atoms stronger and shorter compared to the longer and weaker single bond between the carbons in Sil B. So because of these differences, we will be talking about the hybridization and free rotation, which is related to the Lewis structure of these related molecules. So this figure represents both Sil A and Sil B. The dotted line represents a double bond that exists in Sil A, but also does not exist in Sil B. So upon closer examination, we can examine the hybridization of Sil A versus Sil B. And just as a review, hybridization is an intermixing of atomic orbitals of different shapes and nearly the same energy to give the same number of hybrid orbitals of the same shape and equal energy and orientation that were combined. And this is to minimize repulsions between these hybridized orbitals. And this diagram represents sp2 hybridization, where 3 orbitals are formed and there is 1 additional unhybridized p orbital. However, if we had sp3 hybridization, we would have an additional hybrid orbital and no remaining unhybridized orbital. So looking back at the structure of Sil A and Sil B, every atom has a unique number of bonds and lone pairs, and the hybridization of each molecule determines the total number of sigma bonds and pi bonds. And this is why, and the presence of pi bonds eliminates free rotations around a bond. So Sil A has 6 pi bonds, while Sil B has 7 pi bonds. And for each double bond, there is 1 pi bond and 1 sigma bond. And for single bonds, there is only 1 sigma bond. So free rotation is not allowed in double bonds because the free rotation would increase the repulsion among bonds. So free rotation is not allowed in double bonds because with free rotation, there's a possibility that you end up with 2 out-of-phase p orbitals that creates nodes where there is 0 electron density, and you are unable to form a pi bond. Instead, you need to make sure that the p orbitals are always in phase to ensure that they form a pi bond, and therefore free rotation is not allowed. In Sil B, there are 8 carbons which are sp3 hybridized, which means that there are 4 hybrid orbitals and no unhybridized p orbital that can form a pi bond. Sil B, there is an additional 7 carbons which are sp2 hybridized, which means that the carbon has 3 hybrid orbitals and 1 unhybridized p orbital, which can be used to form a pi bond. Just as a reminder, sp3 hybridization means that you have 4 hybrid orbitals that are degenerate, which means that they're the same energy level, and you have no unhybridized p orbitals that you can use to make pi bonds. sp2 hybridization, however, means that you have 3 hybridized orbitals and an additional unhybridized p orbital. And this on a carbon would be your 2pz orbital. For Sil-A, there are only 6 carbons that are sp3 hybridized. However, there are 9 carbons that are sp2 hybridized. The increase in carbon sp2 hybridization that we see in Sil-A accounts for the double bond that we see here. With less hybridized orbitals, the carbons have additional 2pz unhybridized orbitals that can interact with another unhybridized p orbital to form a pi bond. In the case of Sil-A, the 2 unhybridized 2pz orbitals from each carbon interact in a sideways manner to create pi bonds. Since there are other sp2 hybrid orbitals in both Sil-A and Sil-B, resonance is possible. However, these resonance forms are not major contributors because they raise the formal charge of various atoms. If there are only sp3 orbitals in Sil-B and Sil-A, there would be no pi bonds or resonance forms because you need an unhybridized orbital to form a pi bond. So free rotation is possible when there are sigma bonds and no resonance because out-of-phase interactions between bonds, which inhibits free rotation, does not exist. Hybridization accounts for pi bonds, the ability to form double bonds, and the molecule's ability to form resonance structures. In Sello's experiment, the double bonds are critical to the selectivity of inhibitors because you don't want an inhibitor to target H20S instead of the MTB20S. So syringulin is a molecule and its molecular structure can tell us a lot more about selectivity. Sil-A is less saturated with hydrogen atoms, but it contains an additional double bond. Therefore, there's a greater electron density in that area, which makes the molecule more reactive. However, this increased reactivity also makes it less selective than Sil-B, which, as mentioned before, is more saturated with hydrogen atoms and has a single bond. And these kinds of patterns are useful for future research in order to find other inhibitors with the greatest selectivity. So on to the summary and conclusion. In the summary of the experiment, Mycobacterium tuberculosis is a dangerous and life-threatening disease that scientists have been trying to find ways in order to control its growth. And one of these ways is the creation of inhibitors that can react with its enzymes. The reaction that is formed is a chemical reaction which involves syringulin as a reactant. And the Struck-Lewis structure of syringulin and its hybridization and number of pi bonds and sigma bonds is important in determining its reactivity and also its selectivity.